Hi everyone, welcome to the part 2 of our first chapter, which is cell death. So, the first topic here is apoptosis. Apoptosis क्या होता है? Apoptosis is a type of cell death which is tightly regulated suicide program. So it was necrosis. It was not predefined, but apoptosis was predefined. It is a suicide program which is tightly regulated. In this, the cells were destined to die. So those cells activate their intrinsic enzymes and then they degrade the cell's DNA and the nucleus and cytoplasmic proteins, and then ultimately the cell is dead. So now these cells are converted to apoptotic bodies, and these apoptotic body have portion of both cytoplasm and nucleus. So this apoptosis can occur without it is occurring without any inflammation because the cell is eaten before the contents even leak out. So this apoptosis can be physiological or pathological. In physiological, we have that with development there is decrease in the number of cells, so that is by apoptosis. Then apoptosis occurs after hormone withdrawal, as in menopause. Afterward, it occurs in cell turnover in increasing population. So what does this means? Whenever any immune reaction occurs, there are some immature lymphocytes which fail to express the receptor, so they are apoptosized. Afterwards, there is apoptosis of self-reactive lymphocyte. Then there is death of host after the purpose, as there is apoptosis of lymphocyte after the immune response is done. So next we have pathological causes. First is during DNA damage, then when the misfolded proteins are increased, and then in the infections. Infections can be viral. In viral infections we have HIV, and then it can be host immune response, which we have hepatitis. Another pathological causes pathological atrophy after the duct obstruction. So whenever there is obstruction of duct, it will lead to cell death by apoptosis. So this was about apoptosis, a brief overview. So necrosis was never supposed to happen, but apoptosis is supposed to happen, and it is tightly regulated suicide program which can occur both physiologically and pathologically. So what is the basic mechanism of apoptosis? The cells which have to die will activate their intrinsic enzyme, and those enzymes will degrade the cell's DNA and the proteins and ultimately lead to cell death. Now let's see the morphological feature of apoptosis through the images. So this first one is the epidermal cell. In a normal cell, we see tightly packed organelles. Now this epidermal cell is undergoing apoptosis. We see cell shrinkage, dense and eosinophilic cytoplasm. So the cytoplasm is dense and eosinophilic, and we see a brightly red cuvette bodies, which is the apoptotic cells of skin. So this is an epidermal cell. We can see the cell has shrunken in the size. It is dense and eosinophilic cytoplasm, and this has become an apoptotic body known as the cuvette body of skin. This is the second image. In this this is the electron micrograph. So here what we can see, that there is dense mass of various shapes and sizes around the cell, and we can also see chromatin aggregates peripherally. So this is the electron micrograph of the apoptosis. Okay, so these are other stains. Now this is under phase contrast microscope. We can see that this apoptotic cell undergo blebbing and forms apoptotic body. So that was apoptotic cell. This is the apoptotic bodies. This is under the stain for DNA. So we can see a fragmentation, and this shows the presence of caspase 3. So this is all about the morphological characteristics of apoptosis. In skin, we can see the cuvette bodies, which are apoptotic bodies of skin and which has shrunken in the size, has dense eosinophilic cytoplasm. Then here we can see chromatin aggregates at peripheries, and there are various shapes and sizes, some material. And here we can see that apoptotic cell undergo blebbing to form apoptotic body. This is chain for DNA. Here we can see presence of caspase 3. Before diving into the explanation for a pathway, let's do some basics. So apoptosis is induced by the caspases. So what are the caspases? Caspases are serine proteases. So these caspases cleave after aspartic acid residue. So these caspases, these enzymes, stimulate the apoptosis to happen. Now this apoptosis has two phases: initiation and execution. And the pathway of apoptosis can be intrinsic or extrinsic. Intrinsic is known as the mitochondrial pathway. Extrinsic is known as the death receptor pathway. Okay, so before moving into the detail, let's do what is Bcl. So Bcl is a family of protein which is used in the apoptosis. So there are anti-apoptotic proteins, there are pro-apoptotic, and there are regulated apoptosis inhibitors. In the anti-apoptotic proteins, we have Bcl2, Bcl-XL, and MCl1. So in anti-apoptotic proteins, we have BH1 to 4 subunits. And so what is the function of these anti-apoptotic proteins? They keep outer mitochondrial membrane impermeable. Okay. So pro-apoptotic are backs and back. They have BH1 to 3 subunit, and whenever they are stimulated, they oligomerize and increase the permeability of outer mitochondrial membrane. Then there are regulated apoptosis inhibitor. They are BAD, BID, BIM, PUMA, NOXA. They have BH3 subunits. So this was about apoptosis, which is initiated by caspase, has two parts, initiation and execution, has two pathway, intrinsic and extrinsic. Then we have Bcl family of proteins, which are of three types: anti-apoptotic, which keeps the mitochondrial membrane impermeable; pro-apoptotic, which increase the permeability; and we have regulated apoptosis initiators. So let's start the mechanism. Firstly, we'll see the mitochondrial or the intrinsic pathway. Whenever there is any cell injury, like there is growth factor withdrawal, there is DNA damage, or there is protein misfolding, what happens? It is sensed by Bcl2 family sensors. So now the sensors will tell Bcl2 family effectors that there is a cell injury, which is Bach's back. So what is this Bach's back? It is pro-apoptotic factors. So what they will do, they will go to mitochondria and increase its permeability. So when the permeability is increased, cytochrome C and other pro-apoptotic proteins will leak out. So now these proteins will activate the initiator caspases, and these initiator caspases will activate the executioner caspases. So till now, after this we will discuss after doing the death receptor pathway. So what we did till now, in the mitochondrial pathway, whenever cell injury happens, Bcl2 family sensors sense this, they tell effectors and they increase the permeability. Cytochrome C and pro-apoptotic proteins are leaked out, which stimulate the initiator caspases. So now what are these regulators? They are Bcl2, Bcl-XL, which are the anti-apoptotic factors. In normal situation, they keep this inhibited. So now the death receptor pathway. What happens in this? This cell has Tnf receptor, and this cell has some receptor which bind to its ligand, which is Fas or TNF receptor. So now this is the death receptor pathway, where this receptor ligand interaction will stimulate the adapter protein, which will stimulate the initiator caspase, and they will stimulate the executioner caspase. So ultimately both the pathways stimulate executioner caspases. So now what happens when the executioner caspases are stimulated? first of all there will be endonuclease activation, which will lead to nuclear fragmentation. Then there will be breakdown of the skeleton. Ultimately what happens, we see a cytoplasmic bleb, and when it is broken off from the cell, it is known as apoptotic body. And then this apoptotic body has the ligand for the phagocytic cell receptor, and then the receptor bind with the phagocytic receptors and leads to the phagocytosis. So that was a basic mechanism of apoptosis. We saw two pathways, mitochondrial and the death receptor pathway. So now let's see another intrinsic pathway of apoptosis in some detail. So this is a viable cell. Normally this viable cell gets some survival signs, like it has growth factor, which leads to production of anti-apoptotic proteins, which therefore this mitochondrial membrane remains impermeable and there is no leakage of cytochrome C or any pro-apoptotic factors. So what happens in apoptosis? There is either lack of the survival agents or there is any cell injury like irradiation, which will lead to DNA damage. And there can be increase in the misfolded protein, which will lead to ER stress. So these three things, any of these three things will lead to activation of sensors, which are a BH3 only protein. So what these will do, these will antagonize Bcl2, which was anti-apoptotic factor, and it will stimulate the pro-apoptotic factors. So what will happen then? It will lead to activation of back channel, which will further inhibit our Bcl to BCl-XL. So when this is activated, they will form oligomers that will insert into the membrane and increase its permeability. It will lead to leakage of the proteins, which will lead to activation of caspases and ultimately apoptosis. So let's dive into some more detail. What happens in the cytoplasm? The leaked cytochrome C bind with APAF1, which is apoptosis activating factor 1. These both will join to form apoptosome. It will bind to the caspase 9. So this caspase 9 is the initiator caspase of the mitochondrial pathway. So when it binds to the caspase 9, it will lead to autocatalytic cleavage, and this will lead to generation of catalytically active form of enzymes, and this will again lead to a cascade of all the caspase activation, and then they will mediate the execution phase. Okay, so what happens in apoptosome is formed, which binds to caspase 9, leads to autocatalytic cleavage, activation of enzyme, other caspases are activated, and then execution phase is executed. So what happens, one more thing, that there are some mitochondrial proteins like SMAC or Diablo. They bind to the cytoplasmic proteins and neutralize them, and they normally inhibit the initiation phase of apoptosis. But what happens in apoptosis? These are also inhibited. Okay, so we saw the intrinsic pathway. Now we'll see the extrinsic pathway. So what we see here is two cells. This first cell is the T cell that recognizes self-antigen, or it can be any T cell that kill virus infected or human cells. Okay, so these cells have a ligand known as Fas ligand. And what is this? This is our mitochondrial membrane. Okay, so this has a Fas receptor. Initially what is there is only one Fas receptor, and then they come together and they join and form a polymer, and then when they join, they form binding site for FADD. It is Fas associated death domain. So when it bind to FADD, procaspase 8 is stimulated. It will make it active caspase 8, and then it will simulate the executioner caspase and lead to apoptosis. So now what was this? So there are Tnf receptors family, and Tnf receptors which are under the family of plasma membrane death receptors. Okay, So these are the TNF receptors. This death domain is a cytoplasmic domain, and it works by protein interaction. Okay, so what happens when this interaction is not present? It will trigger the inflammatory cascades. So this was about the extrinsic pathway of apoptosis. So now this extrinsic apoptosis pathway is inhibited by a protein named FLIP. FLIP bind to caspase 8, block FADD binding, and so some virus and normal cells produce FLIP and inhibit the apoptosis. So now we will see the caspase pathway. Intrinsic pathway has caspase 9 as the initiator. Extrinsic has caspase 8 and 10. These both will stimulate executioner caspase 3 and 6, which will cleave inhibitor of the DNase. So DNase will be activated, which will lead to DNA degradation. These executioner caspase will proteolyze the structural components of the nuclear and matrix, so the proteins are also dissolved. It all leads to apoptosis. So now we will see removal of the dead cell. In the apoptotic cell, there is a phosphatidylserine molecule which is normally inside the cell. When it undergoes apoptosis, it flips out, and this is recognized by the macrophage. They will produce a protein that will bind to the apoptotic cell and lead to its engulfment. Sometimes the apoptotic bodies may be coated by natural antibodies and the complement system, which are recognized by the phagocytes, which will lead to its phagocytosis, known as the efferocytosis. So what happens? The pro-inflammatory cytokines are reduced in the macrophage that have ingested apoptotic bodies, which will limit the inflammation. Now let's see another mechanisms of cell death. First is the necroptosis. As the name suggests, it is combination of necrosis and apoptosis. So how does we say it as programmed necrosis. Morphologically it resembles necrosis, and biochemically also it resembles necrosis, but its mechanism resembles apoptosis. So how it resembles apoptosis? It is stimulated by signal transduction pathway, but it is caspase independent. And how it resembles necrosis? There is loss of ATP, and there is generation of reactive oxygen species. There is release of lysosomal enzymes, and there is rupture of the plasma membrane. So it can occur physiologically and pathologically. Physiologically, it occurs during formation of mammalian bone growth plate. Pathologically, it occurs in steatohepatitis, acute pancreatitis, ischemic reperfusion injury, and Parkinson disease. It is a backup mechanism in the host defense against certain viruses that encode caspase inhibitors. So as there is no caspase here, so it can occur in the disease where the virus encode caspase inhibitor. One such virus is cytomegalovirus. So this was about necroptosis, which is morphologically resembled by necrosis, and mechanism resembles apoptosis, but it is caspase independent. So let's see the mechanism. So we have this TNF receptors, and we get this TNF molecule. So what is the starting? There is ligation of receptor with the ligand. So the ligand is TNF, receptor is TNF receptor 1. After that, there is receptor interacting protein kinase 1, which is stimulated, and it forms a complex which is RIPK1 complex, and then it will join with FADD molecule. It will lead to oligomerization of MLKL proteins, and then they will translocate to the plasma membrane and lead to plasma membrane disruption, and then the ultimate result is tissue damage, cell death by necroptosis, and inflammation. So this is same as necrosis. This mechanism was same as apoptosis. So that is the mechanism of necroptosis. Another mechanism is pyroptosis. Pyro means fever, ptosis means apoptosis. So in this type of apoptosis, there is release of fever-inducing cytokine, which is interleukin-1. So the microbial products will enter the infected cell, which are recognized by the cytoplasmic innate immune receptors, which will stimulate inflammasome and further stimulate caspase-1. So what is this caspase-1? It is interleukin-1 beta converted enzyme. It will stimulate a precursor and convert it into interleukin-1. So this is a mechanism by which microbes cause death of the infected cell plus some local infection. Next we have pharaptosis. So pharaptosis is when iron or reactive oxygen species overwhelm glutathione dependent antioxidant defense. So when the antioxidant defense is decreased or is won over by reactive oxygen species or iron, they will increase membrane lipid peroxidation, which will disrupt the membrane function, which will lead to the loss of permeability and ultimately lead to cell death. So it resembles necrosis, but it is regulated by the signals and it can be prevented by decreasing iron. So it is not irreversible kind of thing, but there is loss of membrane crista and there is ruptured outer mitochondrial membrane. It is linked to cancer, neurodegenerative disorder and stroke. The next topic is autophagy. Autophagy, as the name suggests, it is when cell eats own contents and release the cytoplasmic material to the cytoplasm. Physiologically, it occurs in aging and during exercise. So what happens here? The endoplasmic reticulum membrane converts into isolation membrane, which forms a phagopore. So this phagopore is the part of isolation membrane which was derived from endoplasmic reticulum membrane, and this phagopore now makes a vesicle known as autophagosome, which has organelles and cytoplasmic structure. It combines with lysosome to become mature, and this mature thing is known as autophagolysosome. Formation of this phagopore and autophagosome require two ubiquitin-like conjugation systems. So what happens during this process? There is linking of PE. There is covalent linking of PE with LC3, and these compound forms a marker for autophagy. One function of this marker is to target the protein aggregates and evade the organelles. There are some autophagy related genes known as ATGs, so they are also important in formation of this autophagosome. Autophagy is a survival mechanism in stress, and the atrophic cells are exposed to and when the atrophic cells are exposed to decreased nutrients, it is also present or required during the turnover of organelles and clearance of the intracellular aggregates. So this was about autophagy, which is when cell eats its own contents. Autophagy can lead to cell death, but it is not apoptosis or necrosis. So it is implicated in cancer, where autophagy can both promotes cancer growth and it also acts as defense against cancer. It is also implicated in Alzheimer's, where we see impaired autophagosome maturation. It is also implicated in Huntington's disease, where mutant Huntingtin protein impair the autophagy. It also degrades many pathogens such as mycobacteria. So whenever there is deletion of ATG5, it increases the susceptibility to tuberculosis. It is also implicated in Crohn's disease and ulcerative colitis. Here what we see is single nucleotide polymorphisms in the autophagy related gene known as 16L1. So this was all about autophagy.